Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Hello, welcome everybody, and welcome to the restart of the SB Grid webinar series for this year. As you've probably noticed, we are using a slightly different Zoom system, so I apologize if there are any hiccups with the waiting room. But welcome everybody. We're going to be this year we're restarting on what's closer to a monthly basis. So for the next webinar is going to be November 8th. And that's going to be Thorsten Wagner talking about Tomo Twin Cryo-ET. Today, we've got Jan Kozinski, who's a group leader at EMBL Hamburg, and he's going to be talking to us about a new software package for integrative structural modeling called Assembline. And if you have questions as we go through, please feel free to send them to me or Jim in the chat, and we'll moderate questions at the end. With that, Jan, thank you for presenting today, and everybody, thank you for joining us. I will turn it over to you.
Okay, I think I managed to put the slides on.
Yep, slides are on. You're unmuted. And
okay, great. So yeah, thanks first for the invitation, and this opportunity to tell you about Assembline in a bit more detail than I speak usually. I guess most of you are interested in using it and use SB Grid. So I think that would be a great occasion for to tell you how exactly you can run it and what exactly Assembline does. Yes, so Assembline is a software for integrative structure modeling of macromolecular complexes. And to explain you what integrative structure modeling is, I will use this example from our. Give me a second, I need to close this. Okay, so Assembline is a software for integrative structure modeling, and to explain to you what it is, it's a methodology that aims at solving structures of macromolecules like this nuclear pore complex based on low resolution data. So in this example, in 2016, we worked together with Martin Beck at EMBL Heidelberg when I was a postdoc in his group at that time on the nuclear pore complex. And nuclear pore complex is a huge protein complex composed of 30 proteins in 1,000 copies. So it cannot be really isolated and analyzed with methods like single particle cryo-EM. And because of that, also it cannot be crystallized. So the resolution of the data for this complex has always been relatively low. And here, for instance, in the time, Martin's lab managed to obtain a map at 23 angstrom resolution, which was a breakthrough for us that time, but still it doesn't allow to build the structure directly. So what I did that time, I collected crystal structures and homology models of proteins that take part that consist belong to this complex, and also other information, because if you just take these proteins alone, you cannot really fit them unambiguously in this low resolution map. So what we use distance restraints from a technique called cross-linking mass spectrometry. I'll be not explaining what exactly it is, but it's a mass spectrometry technique that allows to measure which residues in a complex are close to each other. So providing some distance restraints for some pairs of residues in the complex. And integrative structure modeling is a methodology, often at least to some extent automated, that takes this orthogonal structural data and proposes models that obtain a precision that is higher than we could obtain by using just one of those techniques or the data sources alone. And that time and this was the first time when actually I started developing assembly. So in 2016, I got this first protocol for modeling complexes just for this specific application to model the human nuclear pore complex. And in 2016, we published this structure, which was quite a breakthrough, but it only contained 30% of the entire complex. And together with Martin, we continued working on this nuclear pore complex, also on nuclear pore complexes from other species. And this year, for instance, we published an update on the structure of the human nuclear pore complex, combining now AlphaFold and assembly together. And just show you the advance. So, well, that time we had a structure that comprised only 35 megadalton of the complex out of 120 megadalton or in total, and now we have 70 megadalton of the complex by combination of AlphaFold and assembly. And over the years, I applied assembly first as a postdoc and then in my group. We applied assembly to various complexes, so many nuclear pore complexes from other species, also from yeast, like you have an entire gallery here, but also other complexes like some bacterial secretion system and this elongator complex. And this elongator complex is particularly cool example, as it shows that actually integrative modeling sometimes gives quite accurate structures and provides a validation. So on the left, you have an integrative model that we built in 2017 using this assembly pipeline based on negative stain electron microscopy at around 30 angstrom resolution, and some crystal structures and homology models. And on the right, you see a cryo-EM structure that was obtained by our collaborators using high-resolution cryo-EM. And you can see that the models are very similar, the architecture is very similar. The functional sites and some domains of in the complex, which I indicate with these labels, are also laid out in a similar way in the cryo-EM structure and the integrative model. So this shows that although integrative models usually have low precision, often they can actually still be used for functional inference and understanding how functional sites are distributed within the architecture of your complex. And so I think basically assembly in the beginning was never supposed to be a software that we would release and maintain and regularly update. But with time, it actually matured. So when we apply this to different projects, we released the code of the scripts before, but then we decided to actually put it together into a more consistent package. So maybe others would also find it useful. And we call this package assembly. So assembly line of macromolecular complexes. Already in my group, after we started here in Hamburg, at EMBA Hamburg, with my two PhD students, Vasilios Rantos and Kai Kaius, who helped me to put this, to package this this software to add some missing features and release as a protocol. So we released that as a very detailed protocol in Nature Protocols this year. And we call it a protocol because it's actually a huge pipeline that combines different software programs from other groups and adds some algorithms on top. And the main engine of assembly is Integrative Modeling Platform, IMP, from Andrei Shari at USCSF, which is one of the main libraries or main software packages for integrative modeling. And it combines different layers. So it does provide some softwares, some standalone programs that you can run for some very specific modeling applications for some simple usage. But in addition, it also provides programming libraries that you can use to implement your own programs or your own scripts, like a Python library and also C++ library. So you can imagine that this is a kind of MATLAB for integrated for structural modeling. And this is how we use it in assembly. So we use IMP as a Python library to implement various features like Monte Carlo approaches and some other things I will show later. And how assembly line works. So first I will show you just an overview with this video. So imagine you have electron microscopy map. So these are low resolution electron microscopy map from negative stain. And then we take structures or homology models of the proteins. We optionally convert them to some coarse grain representation. And then we use some very special algorithm that very quickly tries different combinations of the fits of the structures in the map. And at the same time, but what is not displayed can use other restraints like crosslinks in this case. And this we run this procedure many times, always starting from different random orientations, and then we generate many different models in this first optimization step. And then after we got these models, we go to the second step. Where we select the top models and then refine in the refinement step where proteins can now, so we put the restraints back, so we put electron microscopy and crosslinks back, and then subunits can move a bit more slowly to refine the positions. And then we combine these models, we compare, analyze, we provide scripts for that to obtain some models. And then if you use coarse graining, we also provide tools to go back to the atomic representation after that. So assembly is a pipeline composed of many steps, which I roughly described with this video. I will try now to tell you a bit more detail about these steps. I usually don't present this in such a detail, but I was actually asked in the guidelines for this presentation to also include, you know, even like command line options and how you run this program. So I will attempt that to do that for the first time. So this is the overall pipeline, that figure that you can see in our paper, which now I'll go through. So the assembly usually starts with structures and electron microscopy map. It doesn't have to, so you can run it. You can use assembly also for cases where you don't have electron microscopy map, or maybe just crosslinks. But the most common use case and algorithmic advance that we provide is for electron microscopy maps. And structures, crystal structures, or now AlphaFold models also are the required input. And then in the first step, what we do, we generate so-called fit libraries. So for each structure or each domain, each independent structure that we have, we fit each of them separately to the electron microscopy map to provide a spectrum of possible fits in all kinds of locations, each fit with associated score. How we do it, we integrate yet another program, which for sure you know, it's a UCSF Chimera program, which we use in a command line way, in an automated way, to fit structures into the map. So how it works, we take a structure, then we convert the structure to the density using Chimera tools. And then in this global fitting by Chimera, the structure will be placed in a random position in the map. And then starting from this position, the fit will be locally optimized to fit the density as much as it can. And then we repeat this again placing the structure randomly and optimizing to fit to the density map. And we can repeat this many times depending on the settings by the user like 10,000 or 100,000 times depending on the size of the map. this program can be run easily with one of the programs, a program that comes with Assembline. So when you install Assembline, you get a set of scripts automatically installed, and you start always with this first one, fit.py, which takes a parameter file and runs the fitting for you. The parameter files is a relatively semi-simple Python file where you specify your structures, your electron microscopy map, and also settings for the fitting. And probably you can see that some of the parameters are defined in a simpler way, some maybe in a bit more complex, with some very special Python syntax. And this is more or less the philosophy of Assembline that we have, that you can edit all those files even without knowing Python. If you just replace, you can replace some values here without knowing syntax. But in the same time, if you know Python and you need to modify the protocols for some more advanced usage, you always have access to that, and you can do it. So this first script, the fit.py, would take your structures, all structures that you have, and fit to all the maps that you have automatically. And in the end, we'll return a list of the structures. But in fitting, which is usually guided by cross-correlation, these correlation values are really behaving differently depending on the map, on the quality, and the size of the structures. So in general, how do you know whether the fit is good or bad? If you have, for instance, a cross-correlation of 0.8. And it's actually not clear. And for Assembline to use those fits later, we actually use another trick that we convert these correlation values to p-values that are more comparable with each other. And for that, we assess the statistical significance of the fits, which works like that we take the map and the structure, we run our fitting program with Chimera to create the distribution of fits. So fit lists and scores, and score histograms. We derive all the fits. So we take good and bad fits, entire spectrum. And from this distribution, we estimate p-values for each fit to understand, to quantify roughly what is the probability of seeing a fit that is as good or better than the one observed by chance for the given pair of structure and electron microscopy map. And this is just demonstration how useful it is, even if you don't run the further assembling steps, so that if you, for instance, take this structure and fit to the map, you see that you have very significant p-values, and this structure fits unambiguously in 4 locations. But sometimes when you have a small subunit or small domain, it would fit to the map in many different places in the same way. And you may actually get very good cross-correlation for these fits, but because the cross-correlation values are all the same across the map, the p-values would say that you cannot actually say which one is correct. And this you can run, you can calculate these p-values in the second step that we provide in the second script. We just takes the output directory of the previous step as an input. And this script will create lists of the fits in CSV files with the transformations that you can use in 2 ways. So first, these values can be used in Assembline later on as restraints for fitting in the electron microscopy map. But you can actually also stop here and actually take the fits, use our scripts to generate the fits, the PDB files with the fits. And often actually this is sufficient for many fitting projects that you actually, because if you find unambiguous fits, maybe you don't need the downstream assembling steps. And I would recommend this pipeline even if you don't want to run the full integrative modeling, but you're preparing a paper and you have some fits and you claim they are unambiguous. you could run this pipeline as the first step of assembly to obtain these p-values to substantiate your claim. But otherwise, if you don't get all structures fitting unambiguously or in unambiguous places, you can follow assembling protocol, which then would perform optimization, which I describe in a second. But first, you need to decide whether you want to use coarse-grained representation or not. So for big complexes, if you use atomic representation, and this optimization will just be too computationally intense. So we can use coarse-grained representation, and this coarse-grained representation we provide thanks to IMP in library, and you can actually use a so-called multi-scale coarse-grained representation, where a protein can be represented in multiple ways in the same time. So for instance, here you have a structure represented as one residue per bit. So so each amino acid is 1 bit, like C alpha atom, and also in the same time, each continuous stretch of 10 residues is a bigger bit. And now you can use this representation in modeling in a way that, for instance, for those restraints that need precision and are not computationally too costly, you can use this high resolution representation, for instance, for crosslinks. And for other restraints like electron microscopy fit restraints or in clash score restraint, you can use this low resolution representation. So a balance between the precision and computational efficiency. In addition, you need restraints. So one restraint is electron microscopy map, but you can also include other restraints, for instance, crosslinks or interactions. And for crosslink restraint, we have two main implementation. One is approximating the crosslink as a harmonic spring force with upper bound. So if the crosslink is satisfied below this distance, and if it's above this distance, the proteins will be pulled towards each other. But we also provide a bit more sophisticated restraint, a log harmonic crosslink restraint that converts this standard harmonic, replaces this standard harmonic potential with the log harmonic potential. And this is justified by one of the analyses that we did in collaboration with Michael Nilges from Institut Pasteur, where we noticed that on average, because of flexibility, crosslinks go beyond the maximum physical distance of the crosslinker, and also they approximate log normal distribution. So we can use this log harmonic distribution to approximate this distribution better. So once you have your restraints and structures, and you generated your systematic fit libraries, you can go to run assembline. And assembline takes two configuration files. One is something like system configuration file in a JSON format that defines where you define sequences, subunits, input PDB files, electron microscopy maps, crosslinks. You can also define symmetries and rigid bodies. And the second file is a Python file with parameters that define what type of optimization should be done, what scaling functions should be used for your run, and some options for how to run to adjust the runs to your cluster. And to prepare this, some of those input files, or at least to start, we have this graphical interface in Crosslink Analyzer program or plugin to use this Chimera. So this program has been designed, we created this program to analyze structures in the light of crosslinks, but actually the output of this program or the session of this program can also be used as an input for assembline. So even if you don't have crosslinks, you should actually use this program to start your project. And in this project, you can define the subunits, map them to the chains, or define the chains, and also load the sequence data for each of the subunit and crosslinks. And then, unfortunately, what comes after is a bit of editing in JSON. So to add other parts, at the moment, we don't provide this in the interface. This you have to add manually to the JSON. So you can, from Xlink Analyzer, you can save this JSON file, which is briefly shown here. So without showing the content, which lists the subunits and the data that you load. And then you have to extend this, depending on the project, you add your other directives, like for instance, for symmetry, here for symmetry specification. And this all very well explained in the manual. And maybe at the beginning, maybe it looks a bit scary, that you have to edit the JSON file, but in the end, it's actually very useful. And with some practice, you stop making syntax errors, and actually users are much quicker with this than the graphical interface. The second file for assembly is a parameter file, which is a very basic Python file, which just lists some parameters, like here, for instance, how often the frames of the trajectory should be saved, what kind of representation you want to use, like here it's atomic plus 10 residue per bits, and some other options which I again explained in the manual. So it's a relatively simple file, but it's in Python. So if sometimes when you want to add something more advanced, actually modify the behavior of the program, you can do it in this file. And once you have these two files, which point to all input files, you would run a first step, which is a global optimization. And this global optimization is the key part of assembly that makes it so fast at this stage. So this step would now be taking these fit libraries and generating combinations of them. So randomly drawing fits from these fit libraries and combining them together. And with the Monte Carlo approach, optimizing so they start fitting to each other and also to the electron microscopy map. And because for all these fits, we already calculated the fitting scores, we actually have the p-values, we don't have to recalculate the fits, the fit score to electron microscopy maps anymore during this Monte Carlo procedure. This is all coming in our input table. So this procedure is optimizing the total score that is derived from these p-values and other restraints like crosslinks. And because there's no fit recalculated, this is very fast. And this, how you run it, again, all the complexity is hidden in the parameter files. So it's just a simple command for a single job, and we also provide a command for multiple jobs that would run, in this case, 1,000 jobs or 1,000 different modeling trajectories, and distribute this on the cluster, and you define your cluster submission script in this parameter file. This one, another example how this this global optimization works. So here you have electron microscopy map of the nuclear pore complex and some nucleoporins in this coarse grain representation. And you can see that they immediately jump into the map and do very big jumps and look for the right position and actually converge very quickly. And this is basically thanks to this approach of using these fit libraries, and the program explores the most promising fits very quickly. But in the same time, even though fit libraries were calculated independently, that this step is fitting all of the structures simultaneously. So we run this step many times. It's very rare because of the Monte Carlo stochastic approach. It's very rare that you get the best model in one single run. So we run this many times, depending on the size and the complexity of the complex. You can get a good distribution from hundreds to many dozens of thousands models. And then we provide an optional step of recombination of the top models. So we would take in this step, we take the top models, we retrieve the original fits that were used to construct these models and construct kind of like secondary fit libraries, and then repeat the combinations again. And in the output, often this step provides models that are better scoring than the models that we had before. So kind of reinforcing this entire procedure. And then another optional step is the refinement, in which we would take top models and now optimize the structures within the map without using the fit libraries, but you moving the structures continuously in the map, and every time you move a structure, recalculating the fit. And also there is some support for flexibility of the proteins in this step. And running the refinement is a bit more complicated. You have you need to run a script that prepares the files for the refinement based on the output of the previous step, and then you run, for the moment, quite a complex, at least for the beginner, a shell loop to run all these structures, all these jobs on the cluster. For instance, here refining each of the top models, refining 3 times in parallel. And this program uses the same command as the other steps and uses very similar JSON and parameter file as the previous one. And the last step of assembly is the analysis step, which is quite a big procedure. So I don't put all the commands and I don't go into details because of the time. But this analysis is based on this IMP-SUMCON program. Is a program also developed in SHALI-Lab, which can be connected to other programs that do modeling and allows for all kind of analysis, from clustering to defining the precision of your modeling, defining whether your sampling has been exhaustive, clustering, grouping, and selecting representative models. And this procedure is explained on their website, but we also provide a very detailed instructions on our website how to use it together with Assembline. And the last step, what you can do with Assembline is to prepare files that can be submitted to PDB-Dev. So PDB-Dev is a database which has been designed for integrative models. So in a similar way as PDB works, you can deposit here your integrative models in a special format that, for instance, can also store the coarse-grained representation and links to restraints that were used. And Assembline also uses some components from IMP that format the files for you. And just some highlights of special features we have in Assembline. For instance, this, what we often used for nuclear pore complexes, various membrane restraints. So you can, for instance, devise a restraint that would exclude protein mass from the membrane. So we have a special restraint that where you can define that some protein should not be in that density, so you can expel it from that part. And at low resolution, it often happens that proteins would start penetrating the membranes too much. And also often, actually, we know which domains or which regions of proteins should bind to a membrane. So we also provide a restraint that you can use to anchor proteins to membranes. There's some limited support for flexibility. So Assembline is mostly a rigid body modeling program. So you divide your proteins into smaller fragments, domains, and they are treated rigidly throughout the procedure. So if you want some flexibility, you need to cut them into smaller rigid bodies, for instance, at hinges between domains. But there's also some support for flexibility during this refinement step. But I would recommend it to use only to optimize some linkers between rigid bodies and some little loops. And like here in this case, it helped to refine the position of these rigid bodies because these loops could move. Another cool feature is the multi-state modeling, which is a bit, the moment a bit at the experimental stage. I don't think it's even explained in the manual, but you can actually model several systems in the same time. So if you have different conformations, you can run them together with 3 different maps in the same time. But sometimes at low resolution, if you do it together, if you model different conformations, there will be some random movements between domains, between conformations. And here in this multi-state modeling, you can run them together and restrain them with elastic network, so they are as close to each other as possible within the minimizing these random fluctuations. And just last, some features we added to Assembline to enable this modeling together with AlphaFold, which provided basically much better interfaces, structural models and many good interfaces between subunits. You cannot model many complexes with AlphaFold on its own, so we still kind of need to assemble them together. And this is how it works here for the nuclear pore complex, which Agnieszka modeled. So she took the electron microscopy map from Martin Beck lab and AlphaFold models of different subcomplexes. And then she used Assembline to put it together. And the new feature was a kind of elastic network that you can define, you can derive from overlapping models. For instance, if you have a model of AB, BC, and CD, you can put AB and CD as different rigid bodies, but then restrain the orientation of the 2 rigid bodies by elastic network from the model of BC. So here is what Agnieszka used. She used this global optimization and refinement to build a first model in the electron microscopy map that represented a constricted nuclear pore complex at high resolution. And then she took this model and fit to another map that was obtained in CERN in a dilated conformation. And then she used this refinement step to refine the model into the map. And this, I think, also actually showcases various applications you can use. So you can actually refine the model in the map. So even maybe you already put your structures in the map and you just want to wiggle them and refine so they stop clashing with each other. You can also actually use Assembline just running the refinement step, like here. So the Assembline has been published. So we see it's a protocol. So we published that in a protocol paper, where we also put a lot of the, instructions in the paper itself of how to use it and many detailed explanations. We also have a website which points to the open source code, tutorials, and the manual. The code is here. We put it on the GitHub at the moment, or GitLab of EMBL. At the moment, it's basically a code dump, so we do the development on other branches, but we will be changing this in the near future, so it's more organized. We provide an Anaconda package, which you can use to install Assembline within the Anaconda environment. And SBGrid kindly provides Assembline within SBGrid repository. I must say this website is, this page is very nice, including all the citations that you should use, because in addition to citing, which you don't see on this slide, but you can see it when you go to this web page, because Assembline is a pipeline of many programs. So it's important for us that when you cite Assembline, when you use Assembline, you cite the Assembline paper, but also the other tools that are used internally, like IMP or UCSF Camera. And we list those programs on our website, and SBGrid nicely provides a direct link to that page. So this is just a screenshot of the manual of Assembline. So like a reference manual with all kinds of things you should know about Assembline, configuration files and parameters you can change. And we also provide 2 tutorials, step-by-step tutorials. So one is the elongator tutorial, which is a smaller complex. We can reproduce what we did that time. And this you can run even on a laptop. And this is a relatively small case, and I think very good for start. And we also provide a tutorial that shows how you can model a nuclear pore complex from SRevisier, which is a rather bigger task, and probably you need a computer cluster to run this. Yeah, so just to finish again, acknowledging the people again. So Assembline has been developed over the years by myself, and then Vasilos Rantos and Kai Karius, who contributed and who just recently left the lab, and soon, or actually right now, Dima, who just joined our lab, is taking over Assembline together with me. And the nuclear pore modeling was done by Agnieszka Barska, who is a joint postdoc between Max Planck Biophysics in Frankfurt, Martin Beck's lab at my group, and also other people like Mark and Gerard Hummer's department there contributed to this nuclear pore story. Including the most recent AlphaFold modeling of the nuclear pore. And yeah, thank you for your attention. I would be happy to answer your questions.
Oh, thank you. Again, it still feels weird that there's no applause after a Zoom talk, but you know.
Yeah. Also, I think I must say I actually got used to two in-person presentations, and I was like feeling a bit awkward at the beginning already. So it's interesting.
Oh, so we do have a couple of questions, and if there are more questions, please feel free to send them to me via chat. I'm going to be possibly combining parts of a few here, but I guess one to start with is there a lower size limit for a model or a density map? I would anticipate this might be more of an issue with a small model and a relatively large low resolution density.
Yeah, I mean, yeah, there's no limit of the lower limit. If so, you can do small complexes or even single proteins, right? With Assembline and with this, I think there are sometimes limits when you have a very small domain and a big map, and you don't have much information for the other proteins. Then, of course, and the map is at low resolution. Then you probably Assembline will give you a model where this domain is has a very uncertain position, so it would give you an estimate, and that would be a limitation. But that would be more limitation of the data. The program can run it.
Yeah, and that's going to be empirical and going to depend on the shape of things, and that's
exactly, yeah,
tricky to answer. So I guess you talked about the rigid body flexibility. Could you say a little bit about using flexibility there versus modeling a full length protein as individual domains in the global searching?
Yeah, in the global searching, definitely you need to cut if you have a long protein, and especially now with AlphaFold models, when you have full length proteins modeled, you definitely should cut your protein in small rigid bodies if your analysis of the structure suggests that there is some flexibility. Because that if you have a wrong confirmation, then these fit libraries will not represent what you should get. So this is a limitation of this approach. So you need to have some good guess how to cut into these rigid bodies, and often actually we do it in an interactive manner. That sometimes you based on the results of the full modeling, you actually see that this subunit has a certain position or fits on one side but not the other. In the other models, fits on the other side but not the first, and then you divide into rigid bodies further.
Well, thank you. How would you recommend dealing with potentially unknown stoichiometries if you've got multiple copies? Yeah,
so you can get good impression on stoichiometry sometimes from this first fitting step of the fit libraries. When you analyze these p-values, you would you sometimes see that you have multiple positions with good p-values, and then you can maybe guess stoichiometry from that. Otherwise, you have to assume some stoichiometry, and this is set at the beginning of the program, and then you run the full pipeline, and you see that either you have remaining densities, and then you have to interpret the results and perhaps add extra subunits, or you see that if you have too many copies, then you would see that one protein is jumping around and has a very dispersed distribution, and maybe you need to remove that from the system. So, iterative process.
Sounds as a crystallographer, it sounds very analogous to me to molecular replacement, which I'm not sure how far that analogy goes, but it seems at least conceptually similar. I was reminded of that when you were talking about the p-scores or the p-values for the fitting. It's like this sounds like z-scores.
Yeah, I'm not so familiar with molecular replacement, so I'm not sure whether this analogy holds. But it is in principle, a fitting procedure. So fitting to electron microscopy maps start with the model. I'm not sure whether that's similar to molecular replacement.
Similar enough. It's another search problem to fit the density.
Yeah.
So for making use of models, is there a recommendation for dealing with models that also have things like B factors or AlphaFold reliability scores, PLDT scores?
Yeah, so at the moment we don't use those scores, but it's an excellent idea, and some are programs like Isolde already use that. So for instance, you can use those scores to either to filter some regions, so to remove loops that would stick out in the wrong conformation and cause clashes, or you can actually scale the scoring function that estimates the contacts by those values. So this is definitely one of the features that we want to add. Plus AlphaFold scores, the other scores, the so-called PAE plots, provides very useful information for defining how to cut the models into smaller rigid bodies, which I think is often a step also in the cryo-EM fitting, that you actually now can take those plots and cut your structures into smaller parts, and then you fit them up.
Thank you. And where is it? So one other question, and again, this is one of mine, so it may be a little bit of an ambiguous question, but how do multiple fits for one subunit in like 3 distinct conformations, how do you distinguish that from a non-distinct conformation? Is that something that's clear in the p-value, or is that checking the densities?
So you're asking whether if the same subunit.
I'm just thinking, like, if you have an oligomer with 4 subunits in different positions, is that something that's clearly distinguishable from 4 copies of one subunit that have a poor fit?
Yeah, again, it depends on the differences between subunits and the conformations. So on the shape and the resolution of the map. So I think sometimes you cannot distinguish that if you don't have the data. So if there's nothing in the data that would, if you have a map of 13 angstrom resolution and a couple of different proteins that have very similar shape and no crosslinks on those proteins, that in those cases with the default scoring function that we use, you will not be able to distinguish. Now I think with AlphaFold, maybe there's less of a problem because you can model bigger pieces. But if there's no data, you cannot. If there is a data, it is. So I think assembly provides the mechanism that allows you to do that, but sometimes simply the data is not good enough.
Yeah, at low resolution maps, you're limited by the resolution, and I have spent my share of time looking at them as well. So I do sympathize.
Yeah.
But I keep asking questions, hoping somebody's got a magic bullet, and even though I know better. All right, I think that may wrap things up. So one last call for questions. And Jim, if you've got any questions that have come to you in chat that I haven't seen, feel free to bring them up. But,
there are none.
All right. Well, if not, then yeah, thank you for presenting and for putting all of this software together and packaging it up and making it work. And thanks everybody for attending. We'll see you next month. So.
Yeah, thank you again for listening and the invitation.